Okay, fantastic. Hi, everyone. My name is Shreyans Patel. I'm a fourth year biology student, and my project, it's called a mouthful, Harnessing Chemical Biology, Engineering a Protein Degradation System to Understand the Mechanisms of Cell Fate Decisions. So really quickly, cell fate is important for development and homeostasis. What this really means is kind of starting as early as the zygote or the two-cell stage, your cells kind of develop a polarity, and that kind of determines what lineage they might attend to. So as those cells divide, they might enter the neuronal lineage, they might become blood cells, they might become immune cells, et cetera, et cetera. And so this cell fate is really regulated very closely, and histone chaperones and related proteins have been emerging in recent years as key regulators in maintaining cell fate identity. And one of those proteins is called SHAF1B. And SHAF1B is of particular relevance because it has been previously shown to be heavily overexpressed in certain cancer cell lines. On this slide, I show that we have previous figures from previous studies that show, for example, SHAF1B mRNA is significantly more expressed in leukemia cells as compared to the normal cells. And similarly, in non-small cell lung cancer, a type of epithelial lung cancer, SHAF1B has much higher levels of relative expression compared to similar non-tumorous tissues. And this figure also shows that that's particular to the 95D subtype. So what is SHAF1B? Well, SHAF1B is actually the middle subunit in the CAF1 histone chaperone. And the CAF1 histone chaperone is a heterotrimeric complex made of 3 different subunits that's involved in maintaining cellular lineage identity. And CAF1 is really active during DNA replication, and its job is mainly for histone deposition. And our lab has previously shown that it's really important in maintaining cellular lineage identity by demonstrating that when cells lose CAF1, they kind of lose this ability to maintain their identity. And we've demonstrated that in granulocyte macrophage progenitors, which I'll be referring to as GMPs for short. And when CAF1 is lost in GMPs, they actually shift towards this neutrophil differentiation. They shift towards kind of a more immune system cell. Whereas in mouse embryonic stem cells, when they lose CAF1, they actually shift more towards totipotency, which means they regress towards a more earlier two-cell-like stage. And that's very interesting because currently our lab uses a small hairpin RNA system to achieve CAF1 knockdown. So on the left over here, I have a very simple schematic of how that works. We insert the shRNA gene into the cell, and it's actually inactive to begin with. However, when we present an inducer molecule, the gene can then be transcribed and expressed. And when the shRNA or small hairpin RNA is expressed, it depletes CAF1 through RNA interference. And on the right, I have evidence of that depletion. So on the top right here is immunoblot analysis showing this reduction in CAF1. So as you can see, this SHAF1B band actually goes away as time progresses in 24 to 48 hours. And similarly, we have SHAF1A, which is also involved in CAF1 going away. And on the bottom here, it's again reinforcing the idea that these GMPs shift towards a more neutrophil-like state as they lose CAF1. But one interesting thing to note here, especially in this immunoblot analysis, is that this SHAF1B band doesn't start going away until 24 hours, and SHAF1A actually doesn't start significantly going away until 48 hours. And that leaves a lot of questions unanswered. Specifically, what are the immediate effects due to CAF1 depletion? And we can't answer that using a small hairpin RNA system because it's slow and it takes a while to achieve meaningful knockdown. So actually, the DTAG Degron system presents a solution for studying these immediate effects. Really quickly on the right here, I have a schematic showing this system. So we have a target protein that is fused to this FKBP mutant protein, or what we simply refer to as a Degron. That Degron has a specific binding site that can bind to this degradation tag, or DTAG for short. And when that DTAG binds to this Degron, it can recruit a ligase system, which basically results in the target protein degradation. And this is very fast and very fastly inducible. Altering the DTAG can actually alter the ligase system use, so you can potentially have multiple Degrons in the same system, although that's not the case in my project. And additionally, it's a different method of knockdown, protein degradation, as opposed to RNA interference. And so to apply this to my project, focusing on SHAF1B, we first wanted to test whether this system even works. And so what we did was we did a lentiviral infection into embryonic stem cells and GMPs and kind of introduced an extra copy of SHAF1B with a degron fused to it to see if we could get that to go away. And what I'm showing here is actually a titration. So I supplied DTAG molecule in different concentrations to these cells over 24 hours to see if the SHAF1B fused degron can actually disappear. And what we see here is this HA, which is an epitope that is attached to the degron for easy identification. This band actually goes away in all treatments in all concentrations of DTAG presented. Similarly, this SHAF1B immunoblot also shows this upper band, which is bigger than the endogenous SHAF1B, going away in all 3 treatments. And we have that same confirmation in ESCs with this HA band going away. And so what we learn is concentrations as low as 250 nanomolars in ESCs and 500 nanomolars in GMPs can achieve sufficient degradation. So now that we know the system works, I wanted to go ahead and generate a degron knock-in clone, which means to actually fuse the degron to the endogenous copy. And this was achieved using CRISPR-Cas9 editing. And this is a simple pathway to how we did that. We basically transfected or introduced foreign genetic material into the cells. One plasmid contained the Cas9 enzyme and the guide RNAs necessary, and the other plasmid contained the degron. We seed the cells, select them, and extract single colony DNA. Then we verify them by PCR, or a step known as genotyping. And then we verify that these cells are actually sensitive to DTAG via Westerns. And these red boxes kind of indicate where I am in the project right now. In the future, I want to measure growth defects and conduct maybe RNA-seq or flow cytometry to measure more phenotype. But here's a quick summary of how the CRISPR-mediated homologous recombination actually occurs. So up top, we have the endogenous SHAF1B C-terminal locus. That means towards the 3' end of the gene. And the reason we chose the C term is because we didn't want to edit the N-terminal locus because of previously identified interactions between SHAF1A and SHAF1B at the N-terminal locus of SHAF1B in yeast. So we wanted to avoid messing that up. And so up top, we have the SHAF1B locus, and down here, we have the P-CRIS plasmid containing the degron. So the first thing that happens is CRISPR-Cas9 and the appropriate guide RNAs are actually expressed, and the guide RNAs guide CRISPR-Cas9 to where it needs to make cuts. Once the target sites are reached, Cas9 goes ahead and makes cuts as appropriate, 2 cuts in the degron either side and 1 cut in the SHAF1B locus. This actually leaves a gap in the endogenous SHAF1B, which is actually then filled in, hopefully, by homology-directed recombination. And this process is kind of inefficient, so you have to test a lot of clones. But once we have the clones, we want to genotype them to make sure that we are dealing with clones that are homozygous for the insert, which means both alleles on each chromosome are edited. And so the way we can distinguish between homozygous and heterozygous is by genotyping with primers that flank the degron cassette, in which case, if there's no insertion, you get a 147 base pair fragment, but if there is an insertion, you get a 1057 base pair fragment because of the additional 910 base pairs we inserted. And so on the right here is the first gel that I had from genotyping. I genotyped a total of 24 clones. I have more in culture currently. And I've labeled the clones that are potential candidates as they only carry the insertion and are missing this endogenous band that would otherwise be present in the wild type or the unedited version. And all of the clones have at least one band of the insertion, and that's expected because they're all been selected for that band. However, what's interesting is they also have this anomalous band at 600 to 700 base pairs that's not predicted to be there by genotyping, and I'll touch on that a little bit later. But when I did test clone 8, which was the most promising clone, we actually see that it is sensitive to DTAG. And what we see here is that clone 8 actually has a bigger SHAF1B protein than the normal ESC, and that confirms that it has that degron knock-in. And as we can see, we can achieve efficient SHAF1B depletion over as soon as 1 hour and continue throughout 24 hours, demonstrated by this HA band going away in all 4 treatment lanes and this upper shaft 1B band going away in all 4 treatment lanes as well. So that was really exciting. And furthermore, it actually doesn't matter if we use V1, DTAG V1 or DTAG V13, different DTAG molecules. It really doesn't matter which DTAG we use. We can achieve this efficient knockdown as soon as 1 hour. And we can even use them in combination to achieve this knockdown. So this was very, very exciting. However, as I mentioned earlier, because of this anomalous band between 607 base pairs, it presented kind of like a hiccup in the project. And so when we gel extracted and Sanger sequenced this band, it kind of reveals the following alignment, where this band corresponds to DNA, which is kind of missing the degron, but has part of it. And so it's not sensitive to DTAG. And this was kind of concerning. So right now I'm trying to troubleshoot that band by conducting reverse transcription PCR to verify if that corresponds to an actual mRNA transcript. And in the meantime, I have a few backup plans in place, which is, if we remember back to my overexpression lines, we have constitutive hairpins that can silence endogenous SHAF1B, or maybe even do CRISPR on the overexpression lines to silence endogenous SHAF1B that way as well. And with that, I'd just like to give a huge shout out to my mentor, Dr. Shalufi, without whom, I really wouldn't be able to do this project at all. I'd also like to give a huge shout out to the Shalufi lab, Ruben, Meijuan, Bryant, especially Ruben and Meijuan, for really being like second mentors to me in the lab and always being there to help me out. And of course, Dr. Murr's lab, who we collaborate closely with, and always being there anytime I had questions. So yeah, big thank you to everyone involved. And that concludes my presentation. Thank you so much.
Great job. Thank you so much. Let's get a round of applause for our speaker.